Hello, everyone. Welcome, members. Wow, so many faces in person. Thank you so much for joining us today, and thank you to everyone joining us online. Some quick housekeeping before I introduce our speaker today. We do want to make sure that folks online can hear your questions, so folks in the room, please use the microphone for Q&A at the end. Well, I'm excited today to welcome our speaker, Dr. Jack Dumbacher, Curator of Ornithology and Mammalogy, who will be sharing all about their research in the Galápagos Islands. So thank you again for being here today, and please join me in welcoming Jack.
Thanks, everyone. It's great to be here, and it's great to see the room so full, so I really appreciate everybody coming and turning out. I'm Jack Dumbacher. I'm Curator of Birds and Mammals here at the California Academy of Sciences, and today I want to talk about some of the work that we're doing in the Galápagos Islands, and work that's been going on for a long time in the Galápagos Islands that's been done here at the Academy. So let's start by talking a little bit about the Galápagos. So the Galápagos is a really special place. It has a fascinating natural history and a flora and fauna that's evolved on the islands, and much of it is endemic and found nowhere else on Earth. There are things in the Galápagos like flightless cormorants. And these are related to the cormorants we have here, but because they don't have to go very far for food and because there's no predators on the Galápagos, they've lost their ability to fly. And they have these teeny tiny little wings, and they waddle down to the shore and they go in and they forage. We also have things like the marine iguanas. Now, iguanas are mostly vegetarian, right? So they eat leaves, but it's very dry, especially on the coast of the Galápagos. And so these iguanas have learned to live on seaweed, and they wander out into the water, which is usually pretty cold, and they feed on seaweed until they get too cold to forage anymore, and then they come back on land and sit on that black lava and warm back up again before they can go out. And then also the Galápagos penguin. So this is a species of bird. It's the only penguin that's found in the tropics. And it's partially because of the very cold water that they have there in the Galápagos, and also because there aren't any predators there. We also have things in the Galápagos like giant tortoises, and this is the Santa Cruz giant tortoise, and I'll talk a little bit more about them in a minute. But all kinds of interesting things that you may not have heard of, like daisies that made it to the Galápagos and then have evolved to fill so many different niches, including large, tall, woody trees. So some of the high forests on the mountaintops in the Galápagos are these scalesia forests that are basically daisies. And another thing that's really interesting about the Galápagos, in many islands, when they were first visited by humans, the species on those islands aren't afraid of people. And so you can get pretty close. And that's still true on the Galápagos today, partially because they haven't been hunted. Most of the species haven't been hunted that much. And there's a lot of protections in place right now, so you can get up close and personal to a lot of things. And even Charles Darwin, who was the first scientist to visit the Galápagos in 1835, he wrote about the extreme tameness. He says it's common to all of the terrestrial species. A gun here is superfluous. With the muzzle of the gun, I pushed a hawk off the branch of a tree. And so because of this, we can walk right up. You don't even need a long lens to do photography. These are pictures of a blue-footed booby I took on the nest and wandering albatross that we were able to watch do their breeding dances. So it's amazing to get that kind of look at natural history in different species. So the Galápagos is an archipelago located about 900 kilometers off the coast of Ecuador and about 550 kilometers from the nearest island that's called Cocos here. Because of that, they're very isolated. There are about 15 major islands and many, many more small islets, rocks, and seamounts. But each of the islands has a different size, a different elevation, a different age, different conditions, and this has led to different habitat types. And so each island is like an independent replicate for evolution and for ecology, and also for community assembly. And so the Galápagos have been viewed as a laboratory for evolution. And Charles Darwin was one of the first people to recognize this. And he even said, by far the most remarkable feature in the natural history of this archipelago is that the different islands, to a considerable extent, are inhabited by a different set of beings. I never dreamed that islands about 50 or 60 miles apart, and most of them in sight of each other, formed precisely by the same rocks and placed under the same climate, rising to nearly equal heights, would have been so differently tenanted. And this has been this was the big breakthrough for Charles Darwin. So when he got to the islands, he noticed that there were different radiations of tortoises, for example. And in fact, this fact was first called to his attention by the vice governor, who told him that the tortoise differed from the different islands and that he could with certainty tell from which island any one was brought. And this shocked Darwin because he thought, well, why would a creator need to create a different tortoise for every island? And so not only the tortoises, but also the mockingbirds were one of Darwin's muses, because he visited 4 different islands and he encountered 3 different species of Galapagos mockingbirds, 2 of which are shown here. And he realized that on all of the mainland of South America that he visited, he only saw what looked to him like one form of mockingbird. And yet on the islands, he saw 3 different kinds on 3 different islands that he visited. And this got the wheels spinning, and he started thinking about this. And he realized even then that these were all from the same group, from a same family, and maybe even had relationships or similar ancestors from the past. But he wondered, why would they be different on these different islands? And this was, eventually to lead to his breakthrough and his understanding of how natural selection worked. But it's not just those species. There's so many species that do the same thing on the Galapagos. I've already mentioned the Scalesia, and these are basically daisies that got to the Galapagos. And then once they got there, they radiated into these different forms, some that are scrubby, some that are like tall trees, and some that are, smaller daisy-like plants. And then, of course, the very famous ones that I'm sure you've all heard of are the Galapagos finches, aka Darwin's finches. And, so many people have worked on this, but their situation is so much more complicated than the tortoises or than the mockingbirds that this wasn't really the inspiration for Darwin. And even when he brought these back, it took a while for people, a long time actually, many, many decades, to sort out how many species of finches there were and how they were related to each other. But because much of the flora and fauna of the Galapagos is still intact and present on the islands, and 97% of the landmass in the Galapagos is enclosed in national parks and protected, and because the archipelago provides such an amazing natural history for studying ecology and evolution, a huge number of scientists have worked in the Galapagos over the years. Many classic studies of evolution and ecology have featured Galapagos species, and a huge number of visitors come annually to visit. The number right now is about 300,000 people per year visiting the Galapagos. And you can still get up close and personal with many of the species, which remain quite tame. You have to be careful in some places. There are signs that say, do not touch the breeding birds, which seems crazy, but you actually have to tell people not to do that. So the Academy actually has a long history of working in the Galapagos. And in 1905, the Academy put together 8 young scientists, a navigator, and a cook, shown here in this picture. The youngest person was Gifford, who was only about 18 years old. They purchased a ship, a gaff-rigged schooner, that here is in the port of San Francisco, and they equipped it for a long expedition. And they sailed out of San Francisco on the Schooner Academy on June 28th, 1905. And they spent a year and a day in the Galapagos. And in addition, on the way down, they collected on the Cocos Islands, on Socorro and the Revillagigedos, Clipperton, and other islands off of Mexico. The goal of the expedition was to document what was on the islands of the Galapagos before it was all driven extinct, and perhaps to save it. So even then, they knew that the Galapagos was in trouble, and it was highly impacted because sailors had been dropping goats and rats and cats and different things on the islands. And he knew that they had known that they'd been collecting a lot of the Galapagos tortoises. They probably didn't have the same sorts of notions of conservation as we have today, but they clearly considered this a race to document the flora and fauna before it went extinct. And they clearly saw that establishing these baselines was going to be critical for not only understanding the natural history, but also for preserving the flora and fauna. And this shows the course that they took. So many of the larger islands, they were actually able to visit twice in multiple seasons. So prior to this, they had photographs like this one taken by Rollo Beck, who was our trip leader. And in these photographs, Beck documented the destruction of tortoises by whalers and seafarers. And in fact, scientists today estimate that over 200,000 tortoises were estimated to have been taken from the islands for meat. And most of these were the full adult of the species. And so, and these were taken because you could take these tortoises and put them on their backs in a ship's hold, and they didn't need any food or water for months at a time, and they would stay fresh because they were still alive, and then they could be slaughtered for meat whenever they wanted. And in fact, even in Darwin's time, in 1835, Darwin told stories or heard stories from the local Spaniards that lived on the island that that some ships had come and collected between 400 and 500 tortoises just for the one ship. And that in one afternoon on some of the islands, they could collect as many as 200 tortoises if the men went ashore and were able to collect. So it's amazing how many of them they were taking. They didn't know it at the time, but many of the tortoises, even though they were reproducing, the goats and the rats and the cats and the other things that were on the island were eating the babies as quickly as they were laying eggs. And so, even up to the time that Beck was there were very, very few young on any of the islands. And later than that, there were almost no young to be found on any of the islands. So as they were taking them off, they weren't able to reproduce themselves and replace themselves. Now, we did collect a lot during that expedition, but just for comparison, Cass, which has the largest collection of Galapagos tortoises anywhere in the world, only has about 300 total Galapagos tortoises in our collection. And again, those were meant to document the species that were on the Galapagos. So during this work on the Galapagos, they collected birds and mammals and lizards, tortoises, plants, the geology, the paleobiology, mollusks and insects, and anything else that they could get their hands on. And they went all over the islands. Here's a picture of one of the guys trying to grab a marine iguana. Here, this is Beck, who was the trip leader, along with Hunter, and they're surveying one of the bird colonies and collecting eggs and adults. Here's also a picture of one of the land iguanas and one of the Galapagos mockingbirds that they took during the expedition. And this was ridiculously hard work. I can't imagine any of my colleagues today being willing to go on a ship for a year and work this hard in the tropics with no air conditioning. Here's a picture of some of the guys, and it took multiple men to tie up one of these tortoises and then to carry it down to the shore across lava fields and spiny forests and through cactus gardens. So this was really difficult work, and right on the equator in the heat of the sun. Here's one of the other researchers, probably Slevin, trying to grab a marine iguana and free it from the rocks that it was holding onto. And just to show some of the conditions that they had, this is one of the men's bunks, and this is Rollo Beck piled in. It wasn't even long enough for him to have his legs out, and you can see there wasn't a whole lot of headspace down there. The men also, they were in the Galapagos for over a year, and about a year and a half all told, and they had to fix everything on board. They had to be each other's doctors and nurses. By the end of the trip, their clothes were all falling apart, and we have pictures of them sewing each other's boots back together and sewing each other's pants back together. And the conditions were really difficult because of the heat. Here's a picture of Williams, who was on shore and had gotten lost and came back to the boat just totally scorched and overheated and, suffering from needing water, and was really, really thirsty. And they got some pictures of him when he came back. He was very happy to see the boat. And many of the islands had almost no shade, so they would all try and find a little spot under a cactus to cool off when they were working on shore. So it was really, really difficult work. And in addition to all of the specimens that they took of the animals and plants, they also were equipped with a state-of-the-art camera, and this is Rollo Beck trying to get some photographs. And all of the men on the ship also kept detailed logs and detailed journals of where they went, what they saw. And so some of the first behavioral descriptions, you may have heard of the woodpecker finch on the Galapagos, which actually breaks a spine off of a cactus and uses it as a tool to get grubs out of the bark of trees. And that was first described by Gifford. And this is one of Gifford's pages in his notebook here. So we have amazing specimens from the collections, the archives from the collections, the photographs from the collections. And when they finally did return to San Francisco, they had over 75,000 specimens that were returned to the Academy. This was the longest, most interdisciplinary and expansive expedition ever launched in the Galapagos. And however, the Cal Academy collections go well beyond this one expedition. We also have specimens that were collected by the Hopkins Stanford, which were collected by Stanford in the Hopkins Stanford collections from 1898 to 1899. We also have the Templeton Crocker expedition of 1932, and that was largely launched to complement the specimens that were collected in 1905 and 1906. So they were focusing on marine invertebrates and fish and things in the water. There were also collections made during the 1910s, 1920s, all the way up through the 1950s. And in the 1960s, Bob Bowman, who was a professor at San Francisco State, had been studying Galapagos finches and other birds and insects. And so he made extensive collections, which we also have here at the California Academy of Sciences. And he helped found, as well as many of the people who were here at the Cal Academy, they helped found the Charles Darwin Foundation and the Charles Darwin Research Station on Santa Cruz. So now, today, most of this work is carried on the islands by the Charles Darwin Research Station, which we have close ties with. So altogether, CAS has the most extensive collections, scientific collections of the Galapagos anywhere in the world. And for many taxa, we have more of those things than all of the other museums in the world put together. So it's an amazing collection. It's amazing because a permanent, intangible, and most complete record of life on Earth. Many of the specimens were collected long before we even knew what DNA was, and now we can get DNA from those specimens. We can also get stable isotopes, we can get environmental contaminants, and so that baseline data has proven critical for many of the studies that we do today and critical for some of the conservation efforts that we're involved in, which I'm going to talk about next. And most of what we know, or we think we know about the Galapagos flora and fauna, has its foundations in these specimens. So it's very, very important stuff that we have. There's one other reason why why our 1905-1906 expedition is so important, and that is because while they were away in 1906, the Academy was hit by an earthquake, the 1906 earthquake and fire, and we lost the rest of the museum. So during the expedition, they realized that they were what was left of the Cal Academy. And some people believe that maybe the Cal Academy wouldn't be here today had we not been on that expedition and had we not had those huge collections that we were in charge of and we had to find a new house for. So there's a lot of important history for the Academy here. Now, I also want to point out that these aren't just dusty old specimens that sit in our basement. We use them all the time in our research. And I'm going to give you some examples of just some of the work that I do in birds and mammals. And I'm going to start by telling a story about this bird here. This is the vermilion flycatcher. It's the most brightly colored and beautiful bird in the Galapagos, if you ask me. And they've been declining on the Galapagos. I have a friend, Alvaro Jaramillo, who also lives in the Bay Area, and he leads tours to the Galapagos. And about a decade ago, we were sitting and chatting and having a cup of coffee, and he said, you know, somebody should do a study of these birds because they're declining and they're gone from maybe half of the islands that they used to be on, and no one's really paying attention to it. And he said, and I know because I have recordings of them from San Cristobal. They look different, they sound different, they act different. He said, somebody needs to do some work to see if they're actually a different species. And we had a graduate student at the time, Or Carmi, who was a master's student at San Francisco State, who was looking for a project to do. And we said, Or, why don't you take this on? And he agreed to do it. Now, in order to understand what's going on in the Galapagos, you actually have to put those birds in the context of the larger species. So they were just considered minor, unimportant variants of the vermilion flycatchers that we find on the mainland. And the vermilion flycatcher is found from California all the way through Mexico, Central America, and into Argentina. So it has a huge range. So Orr had to go and contact museums all around the world to get fresh tissues wherever we could get them. And the only specimens that we had from the Galapagos were from the Academy's collections from the 1890s and the early 1900s. So we were able to get DNA from these specimens from the toe pads. We could take a little tiny sliver and we could extract DNA and we could sequence that DNA and use it in our analyses. So he got specimens from all along the range, including the Galapagos. And then he was able to come up with a phylogenetic tree. Now, this is going to be very hard to read, but basically this is the root of the tree, the ancestor, and you can see that there's a split right here, and this leads to all of the continental forms here. And this group up here are all of the Galapagos forms. So the first thing that we realized was that Galapagos is a completely different group from the mainland, and most certainly different species. And then if we zoom in on that group, so here's the ancestor of the Galapagos group, and even within the Galapagos, there's this group that's on San Cristobal, which is the most distinctive, and this is the one that Alvaro mentioned to me. He said, this is the one that's missing, that's now probably extinct from San Cristobal, and it's very distinct. And he thought that this would be distinct from all the others, but our data was also suggesting that there was another split here. At the time, we didn't think that we had enough DNA data because all of the data that we had came from these specimens. We only were able to sequence a small number of genes, and we only had a small number of samples that we were able to work up. But we did end up publishing this paper, and Orr, the graduate student, was the first author, and the title is The Phylogeography of the Vermilion Flycatcher Species Complex, Multiple Speciation Events, Shift in Migratory Behavior, and an Apparent Extinction of Galapagos Endemic Bird Species. Now, this actually made a lot of people in the Galapagos unhappy. And the reason why is because they've always been able to brag that no endemic bird species have gone extinct in the Galapagos. But our work showed that this one species, the vermilion flycatcher of San Cristobal, is very distinct from the others, and it appears to be missing and probably extinct. But what it also did was it shined a light on this problem, and whole teams of researchers began studying the vermilion flycatchers and began trying to figure out what was going on, why were they in decline, and how can we help them. And so that was critically important, and it marked a real turning point for the species. So we were very excited that it was able to help catalyze some of these conservation efforts. The second part of this story is that in 2019, I was down in the Galapagos and visiting some colleagues, and I met this young student, David Anchundia. And he's one of the students who's been studying the vermilion flycatchers as well as some of the other land birds. And he came up to me and said, "Oh my gosh, I didn't realize that you were the guy who did that research on the vermilion flycatchers." He said, "We're so excited that you did that because now we know that there's 2 species on the Galapagos, and it's really helping us figure out how to manage them." And I said, David, that paper isn't clear. We know that there's at least 2 species and probably more, but we didn't have the sampling of all the other islands. And really, I said, to do this right, we're going to need fresh blood samples from as many species as we can get. There's as many islands as we can get. And I said, if you ever want to do this, give me a call and we'll see if we can put something together. And about a year later, he called and he said, "You know what? I'm starting a PhD program and I really want to do a chapter on the genetics of the vermilion flycatcher." And so the Academy was able to bring him up with funds that we have called the Lakeside Funds, which brings scholars from other countries here to use some of our resources. And he was able to go around to all the different islands where he was already catching and banding birds, and he was able to get fresh DNA samples and fresh blood samples. And there was one specimen, he was in the field during a windstorm, and he was watching a nest, and he watched the nest blow down. And the eggs in the nest all broke on the ground. And he said, "Oh my gosh, this is a terrible thing." He said, "But these are really fresh embryos, and from this, I can get good DNA, and we can actually sequence the whole genome of the vermilion flycatcher." And so his quick thinking, he was able to get those embryos on ice and get them back to the Charles Darwin Research Station and keep them in the freezer. And we were able to bring those here, and we were able to sequence the entire genome of the vermilion flycatcher. We've assembled that, and it's a very, very good quality, almost telomere to telomere chromosomal assembly of the whole genome. And it's exactly what we needed to do the kinds of analyses that David wanted to do. And so that allowed us to do that. So I'm going to walk you through some of the work that David was able to do here with us. So in total, we were able to get DNA from 55 samples, and 38 of these were modern blood samples that David was able to collect from the field. So all the places that are in red are samples that David had collected from blood samples from birds that he was banding. All of the samples that are in yellow are ones that we got from the Cal Academy collections. And so you can see that we were able to sample birds from Santiago Island, where he couldn't find the birds anymore, from Marchena, where they're believed to be extirpated, from Rabida, where the birds don't occur anymore, Baltra, where they don't occur anymore, San Cristobal, and Floriana. So this is almost complete sampling. The only island we were missing is Santa Fe, and I literally just got some samples from the American Museum a couple weeks ago. So we're going to include these, but we're going to publish this paper first. And we also had a sample from North America from Arizona that we were able to include in this. So this is a little bit less messy tree than I showed before, but here's the Arizona bird. And then you can see this is how they split on the Galapagos. So San Cristobal, which is now extinct, is the most distinctive group, and it's most certainly its own species. But also Floriana Island, which is also extinct, appears to be its own species. And then if we dive deeper to the next part of the tree, what we find is that Floriana and Isabella Island also form a distinct species. Then if we look at what splits off of those, Rabida and Pinson Island also form a distinct species, which is still extant on Pinson, even though they're missing from Rabida. These appear to be the same. Santiago and Marchena Island are pretty closely related, but they're each different species on those two different islands. And Pinta Island, where they still exist, appears to be a distinct species. And then Santa Cruz, which is one of the islands that David does a lot of work on, they still exist, but there's literally only about 30 individuals left on that island. So they're critically endangered, and they're really concerned about them. So interestingly, like the tortoises and like the mockingbirds, almost each island or island group has its own species, but they just look similar enough that we didn't realize that there was that much genetic variability underlying those birds. And why is this important? Well, it's partially important because before David's work, everyone was concerned about the Santa Cruz population, and they were planning to get some birds from Isabella and Fernandina to augment the genes, to move some of them to Santa Cruz to help that population out. And now that we have the genetics, we know that would have been a terrible idea. So they're not going to do that now. And also, the island of Floriana is an island that they recently removed all the cats and goats and rats and mice from, and they're hoping to put new birds back on this island, not just the Vermilion flycatcher, but many species. And so this work is going to help us try and figure out which ones might be the most ecologically similar and genetically similar to put back on those islands. So the work turned out to be very, very important. And the work that we're doing in the Galapagos, so David's is a really interesting study, and it's, he's working on those papers right now. The genome paper was just accepted, and his phylogeny and his analysis of genetic variation is in work. But it's not the only work that we have going. We have many of these projects going. So his is just an example of the kinds of things that we're doing. So this is my colleague at San Francisco State, Jaime Chavez. He's Ecuadorian, and he's been working in Galapagos his whole life. And so we're co-advising several different students who are working in the Galapagos. This is Ezra Mendales, and he's been studying the lava heron. And it also was just considered a minor subspecies of what's on the mainland. And Ezra's genetic work is showing that they're very distinct, and that there may be multiple species of lava herons on the island as well. And likewise, Pune, she's a PhD student at Berkeley right now, but she was a master's student with us, and she was studying how the yellow warblers are adapting to different environments, to different upland and lowland environments, which are very different in how much water they have and what kinds of plants live there. So she's been studying that. We have another student studying how Darwin's finches and their bills are adapting to urban environments. And we also have new students who are coming in, and they're going to be, and this is part of a larger project that we call Galapagenomes. And in the Galapagenome project, we got NSF funding, and we're going to be studying and creating new genome assemblies for many of the different bird species that are threatened or endangered, including the lava gull and the Galapagos petrel, and we're hoping to work on the Galapagos martin as well. And these are all augmented by the specimens that we have in the collection, and we're able to sequence those and get full genome sequences as long as we have a reference that we can align them to. And birds are what I know best, so that's what I like to talk about, because I can talk about it more authoritatively. But My colleague just sent me this paper this morning, so I was excited to share this. And this is one of my colleagues, Omar, and he works at the Católica University in Quito, Ecuador. But he was recently in Cal Academy, also on one of those lakeside fellowships, and he was looking at some of our lizard specimens because he studies the lava lizards. And he discovered in our collections a new species of lava lizard on Gardner Island. And he just described that. So the specimens in our collection are the type specimens for that new species. And it just goes to show that, like, even though there's been a ton of biologists working in the Galapagos, there's still so much for us to learn. And every time you look deeper, you find new things, even in a place like Galapagos. So we're really excited about all that work. So where are we going with all of it? So one of the things that I'm so excited about, especially in this age of, we're all worried about the environment and so much doom and gloom and environmental fatigue, and we're all tired, and it's hard to keep our enthusiasm for this kind of work. But in the Galapagos, I feel like it's a really different environment. We often have these kinds of genetic data, and we're saying this has obvious management implications. We can use this for conservation, but we wait and we just don't see that much activity. We don't see it happening. But in the Galapagos, it's just exactly the opposite. The managers are ready to do things. The national park wants to go out there. They want to repopulate the islands. They want to rewild the national parks to the extent that they can. But what they need is the scientific data to tell them how to do it, to tell them which species to move where and which species belong. And so we're really excited to be working with our partners on some of these projects. And we just got a big $2 million grant from the Global Environment Fund and the Latin American Development Bank to regenerate some of the Galapagos Islands. So these are some of our partners: Island Conservation, the Durrell Wildlife Trust, Rewild, University of or San Francisco State University, the University of San Francisco de Quito, which is in Ecuador, the Charles Darwin Foundation, the National Park, and the Hoko Toko Foundation. And we're all part of this big grant, and we're all doing different things. And so I'm going to tell you a little bit about what we're up to and how things are going to progress. So the first thing that you have to do when you're regenerating these islands is you have to remove the invasive species that are the threat. So this is usually on the Galapagos goats, but also donkeys, cats, dogs, chickens, pigs, cats, and I always said cats, dogs, mice, and rodents. So those are the really big ones, and it's super hard work. So some of the groups that are already doing that work are Island Conservation and Rewild. And I was there in October and working alongside some of these groups as they were doing the final work on Floriana Island. So this is one of the older islands, and they were using helicopters to fly baits over, and they've got drones that they're able to monitor, and they can see where rats and cats and mice are moving around. And they've also worked really closely with all of the local people, because this island does have people living on it, to make sure that everyone had enclosures for all of their animals so that their chickens don't get out, their pigs don't get out, but that everything can live really well. And the local people are very excited about this. In fact, we were just back in the Galapagos in February, and all of the plantations that they have, for the first time, the corn has no mice or rats chewing on it. And the tapioca plants that they have, which are usually like 60 to 70% destroyed by rats and things, are all looking beautiful. So the local people, who were at first a little bit concerned about all this work, are really excited now that they're seeing how things are panning out. But this is really tough work, and it's kind of brutal, and it's not the kind of thing that all of us are terribly excited about. But the difference that it's making in Galapagos is absolutely huge, and it's a real game changer. And they've already done a lot of this work on the islands of Pinzon, Rábida, Santiago, Santa Fe, and now Floriana is done. So then the next step is to begin moving animals that used to be on those islands back to the islands where they used to be. And this is the work that the Charles Darwin Foundation, the Durrell Wildlife Trust are really heavily involved in. These are zoo folks. They know how to bring animals into captivity, how to move them, how to acclimatize them to a new environment and release them. But the big challenge is to try and figure out what the complete ecosystem used to be like before humans arrived. And that's where they've asked the Cal Academy to come and join this work and to be involved. So one of the things that we're trying to do is document what species were formerly on each of the islands. And to do this, we're using museum biodiversity data. A lot of the data that we already have in our collections, we're making sure that it's digitized. We can easily search it. We can easily make tables of what used to be on all the islands. But we can also search data from other museums, because the American Museum, the Smithsonian, the Field Museum, Berkeley across the bay, and museums like Tring in London, the British Museum, they all have specimens from Galapagos. And so we're pooling all of that data together. And because we're a museum, we know how to do that. We know how to sort through that data. And we're creating those lists even as we speak right now. The next thing that we want to do is, I've mentioned those archives, the photographs and the field notes from all the explorers. The Academy explorers weren't the only ones to do that. All the folks that used to go on these expeditions and collect for museums kept those kinds of notes. So even if they didn't have the specimen, they did often observe things, and these are often written up. And so we're going to be going back to these early records in the archives to pull that material together to see what other species we can document. And the last thing that we're going to be doing is we're going to be doing new paleobiology work. So we're going to be going to lava tubes in caves. So oftentimes these lava tubes, they form when lava flows down to the ocean. And then when the lava as it's flowing, the outsides cool and harden and form a tube, but the lava on the inside continues to flow. And then when the lava source is cut off, then all the lava flows out the bottom and leaves an empty tube or a cave. But oftentimes what happens is, where there's openings into the caves, barn owls and other predators will nest in those areas. And during the day or the night, depending upon whether it's an owl or a hawk, they'll go out and they'll collect all kinds of rodents and small birds and lizards and geckos and eat them. They'll come back and then they'll regurgitate pellets right there in the cave. So there's these huge mounds of centuries of things that these animals, these predators, have eaten as a documentation of what used to be on the islands. The other thing that happens is that there are cases where these lava tubes, the roofs of the lava tubes collapse. And what happens is that different animals will fall into those, and it's like a giant pitfall trap. And then that also will document what used to be on those islands. So we have a new paleobiologist here at the Academy. She's very excited about this work, and we're planning some of our first expeditions to do some of this paleo work, hopefully this summer, if we can get all of our permits in line. So we're really excited about some of this work that we're doing. And just to give you an idea, we went to some of these paleo caves in October, and then also last summer in August. And it's amazing. Like, here's just a picture of the ground on one of these caves, and you can see all these little bird bones. All these little white things are bird and rodent bones that have accumulated. And here's a case of an almost complete, either a hawk or an owl skeleton on the floor of one of these caves. And here's the entrance of one of these caves on Floriana Island that we had to scramble down into. But this one is a pretty famous one because Dave Stedman, who is a paleontologist, did his PhD working on this cave in the 1970s. And so he's one of the ones who pioneered some of these techniques working on the islands in the Galapagos. All of his work was done on, or most of his work was done on Floriana and not so much the other islands. But even on the island of Floriana, he documented dozens of different species that used to be on the islands that aren't there now. So this is the kind of data that we need in order to inform these regeneration projects so that we know which species to move around. So in addition to that project, I've already mentioned the Galapagenomes project, and this is an NSF-funded project. We have a new postdoc. She was just here at the Academy today. She just is finishing up at UC Davis right now. And our idea with this project is to sequence and assemble genomes from as many different species of birds in decline as we can, so that we can use those for those kinds of projects to figure out how much variation there is, how many species are there, and which islands can we take things from and move them to. So we're really excited about this, and the Academy is a key partner for this work because this is our core work. And this is the kind of expertise that we have and that we've built over many, many years. So it's in our science. It's in the research that we do. It's in our collections, in our archives, and we know how to use these records, and the records that we have are amazingly important for these kinds of conservation efforts. And it's in our mission to regenerate the natural world. It's one of the things we do. It's core to what we do. And remember that Rollo Beck and Joseph Slevin and the others who started that collecting in 1905 and 1906, they wanted to do that work because they were hoping to document what was on the Galapagos so that they could save what was on the Galapagos. And I want to, on one hand, I think, gosh, did it really take us this long to get to where we are? But on the other hand, it's like, but here we are, and we're doing that work, and we're able to leverage all these collections. And I think that they would be really proud of the work that they did. So that's why the Academy is involved. And the Galapagos is an ideal place for this kind of work for a variety of reasons. So islands are very fragile, and so it's a place where we can make a big difference. The island ecosystems are a little bit simpler, so it's a great place to go and do these kinds of things and learn in a more simple environment that we can scale up later. The Galapagos, unlike many island archipelagos, the Galapagos is still relatively intact. So our work isn't as big as it might be in some other places. We have a vast collection and a rich history. We have amazing partners. All those other folks that I've already mentioned are amazing to work with, and I feel like we have all the different pieces of the puzzle assembled and ready to do this work. And even more importantly, each island is like an experimental replicate. So just like the evolution work that we were talking about earlier and how each island is a replicate, this provides us a model of how to do regeneration work everywhere. And we start this on the smaller islands where it's a little bit easier to do, and then we can see what works, we can see what doesn't work, we can adjust our plans, and then we can do it better and keep scaling it up. So this project on Floriana was the first island that actually was inhabited, which added a whole extra layer of complexity, but now we're setting our sights on even bigger islands that we can begin to work on. And the Galapagos are so well known that we know that the work that we do there is going to have impact. So we're excited because, you know, people love the Galapagos, and the work that we do there is just like the work that we do in evolution and ecology. They become classic papers and, you know, how-to kinds of things. So again, this is the list of some of the partners, and we're really right now exploring. We were just down there in February, and Scott Sampson, our executive director, was down there talking with all of these partners in a couple of meetings. We had some 2-day meetings. We went to Floriana, and we're exploring how we can have an even larger footprint in the Galapagos as a larger initiative with all of these folks to try and figure out what we can do next and to prototype some of the methods that we can use to reverse the destruction and to rewild the Galapagos. And I also want to mention, some of you have already picked these up, but you have an extraordinary opportunity to be a part of this and to see the Galapagos with us, to visit these islands and experience the wildlife and meet some of our partners, because we're going to be leading a trip, and we've just resurrected the Academy Expeditions. We're going to be leading a trip there next summer. So not 2024, but 2025. So you have a lot of time to think about it. But it's going to be an amazing opportunity, and we've designed a trip that will take us aboard the Isabella II, and being on one of these ships is one of the best ways to see the Galapagos, because you do all your traveling at night between islands. You wake up in front of a new island, it's beautiful, and then you get a chance to go onto it and see what's there. And you know, the ship that we're going to be on is a nice one. It's not too big. I think it only holds about 40 passengers, so it's pretty cozy. And we'll get to see a lot of the different, you know, the wildlife on the Galapagos, the endangered Galapagos tortoises, the finches, the Scalesia forest, many other endangered species. And we're going to get to see some of the key islands, including Floriana Island and some of the other larger islands. So I think that's going to be kind of fun. But before I close, I also just want to thank the Academy for its century of commitment to this kind of conservation. From the 1905-1906 expedition, and these guys who risked their lives and worked so hard to make the specimens and to bring them back to document what was there, to the decades and decades of people who looked after these specimens, because it takes very little time to lose all of that. In fact, many of the specimens that we do have in the collection are from other collections that have shut down, and now we are looking after those, including the Stanford collections. So we really want to thank the Academy for making that century-long commitment to doing this kind of work and maintaining these specimens. And also to thank the Academy for supporting the scientific work that we do. This is Athena Lamb, who runs the Center for Comparative Genomics, and we welcome, you know, so many of our colleagues from other countries to come and do this work with us. And so, you know, thanks to that commitment to the science as well, in keeping all this going. And finally, I really just want to thank all of you, because you're all members of the Academy. We could never do what we do without your commitment to us, and for all the support that you bring to us, and for all the different ways that you show up, from coming to the seminar today to, whatever annual giving and purchasing your membership and coming to see the Academy floor. So thank you, everybody. And I really appreciate all that you do for the Academy and all that you do to make the kind of work that we do in the Galapagos happen. So, and with that, thank you. We'll turn, and if there's any time for questions, we can take some questions. So I think we have a mic that we're going to try and pass around, so that, because I think that there's some people viewing it live.
We
online.
do. We have plenty of folks joining us online. I have one question.
And I can repeat the question too, if it's.
It's a journey back here.
And that's a blue-footed booby from North Seymour. That was my second trip ever to Galapagos in 2008. And I was about as far away as I am to the folks in the front row seeing that bird.
Hello. First off, that was an outstanding talk, and it was just a privilege to be here today. So thank you for that. I understand why the animals on the Galapagos are different from other places, and why natural selection on a set of remote islands would deliver that result. But I have two questions for you. My impression is that the exaggerated results are more notable in the Galapagos than in other sets of remote islands like French Polynesia or the Hawaiian Islands or the Channel Islands. So one, is that true? And if so, why is that? And why is there also the inter-island variation so notable in the Galapagos compared to French Polynesia, Hawaiian Islands, Channel Islands?
Yeah. So there's a lot packed into that. So why is the Galapagos different, if it even is different, right? And I would say it may not be that different. So, like, for example, the Academy has collections from the Revillagigedos Islands. But most of those specimens are in our extinct case. So there's a Revillagigedos dove, there's a Revillagigedos pygmy owl, there's a Revillagigedos wren. But those islands were pretty small, and when rats and cats and other people got onto those islands, most of those species went extinct. Many of them around the turn of the last century, so the early 1900s. And so a lot of islands did have endemic species and things that evolved only on those islands that are distinct from other places. But it's rare to have islands like the Galapagos that still have so many of those things on them. So I think maybe that's the amazing thing about the Galapagos. Another thing that's interesting about the Galapagos is that it's not a great place to be. The climate is rough, it's hot, it's dry. There's only a couple of islands that have water sources. And so unlike the Hawaiian Islands and the Polynesian Islands, where people moved to those islands thousands of years ago, no one really ever lived in the Galapagos until like 100, 150 years ago. So the damage was done mostly by people who passed through and dropped goats on the island and dropped rats on the island, accidentally. But so the person, Dave Stedman, who did the paleo work on Floriana Island, after doing his PhD, he ended up working all across the Pacific. And he documented huge numbers of species in Polynesia and in Hawaii, largely in lava tubes and other caves, all the way over to Melanesia. And for example, on Hawaii, there were giant geese, there were flightless rails, there were all these huge suites of species that were all wiped out by the Polynesians. And one of Dave Stedman's theses at the end of his work was that, we like to think of, and this is somewhat controversial, people don't like to believe this. I got in a match with our executive director over beers in the Galapagos talking about this. But Dave's real findings were that, we like to think of the Polynesians as being in this beautiful harmony with nature. But to the extent that they're in harmony with nature, it's because they ate everything they could and wiped it all out. And what's left is the stuff that they couldn't catch. And so now there's harmony. And I think I think really the lesson is that with every new technology that humans have, we have new power to exploit and overexploit. And we're going to have to check ourselves at some point, because our technology now is growing so rapidly that, like, we know now that we can even change the climate, right? And if we have to be careful. And there's even documentation on the mainland of other species of bison that the Native Americans might have driven extinct, and giant ground sloths, and things in the La Brea tar pit that are no longer here in North America.
Quick follow-up then. So would South Georgia Island or the South Orkney Islands have extraordinary variation because they haven't been populated?
So okay, so that's part of the story. So but if you have like a single island, that's a long way from anything else, it's hard for species to get there, and then and then there's just one island for things to happen on. What happens in the Galapagos is that there's 15 large islands and still some smaller islands. So you get lots of replicates, and then things can evolve. And once they become different enough, they can move to another island, like the finches or the Scalesia. So then you end up, getting all this complexity. The Hawaiian Islands is a great example. And you may have heard about how the Hawaiian Islands is on a volcanic conveyor belt. So, if you go up to Midway Atoll and Laysan Atoll, those are some of the oldest islands, but they're basically, as they're sliding over the hotspot, they form, they become very big islands, new species move to them, they speciate, they become new species, but then as the islands slip away, then they're no longer building, and then they're beginning to erode and break down and sink into the ocean. And then as that happens, things go extinct, but then those are, more distantly related. So the Hawaiian Islands is another good model, which is very much like what's happened in the Galapagos. And there've been a lot of comparisons. And the Hawaiian Islands is probably a little bit older, because the oldest island in the Galapagos is only about 5 million years old. And if you go, all the way out to the oldest line islands, they're much older, but many of them have sunk into the ocean too. So it's that, geographic complexity and the fact that there's many different islands. And so, things can become a little bit more complex. The other thing is that those are tropical islands. So, they're really great for life. Whereas some places like South Georgia are cooler. There's less time in the year for things to evolve there, and to grow, so all those things, feed into it. And then one of the things that's happened in Polynesia and Hawaii is just that humans have been there for a long time. So some of those unique things have already disappeared. And in Galapagos, many of those things are still there on the island. One of the things that Dave Stedman found, so there is a endemic rat in the Galapagos and an endemic mouse, but it's been driven out of most islands, although it still exists on Santa Fe, maybe one or two other islands. I think Santiago has some of them. But there used to be a giant rat too, which is really interesting, because nobody even knew that thing existed until Dave Stedman did his paleo work. And those were probably, that probably disappeared really, really early. So that might be a little bit of a simplistic explanation, but I think the complexity of the number of islands, where they are, when humans first got there, but all of that stuff conspires to make Galapagos a really fascinating place where you can kind of go and get this view into the past of what, an oceanic island may have looked like thousands of years ago when humans first got there.
Hi, I have a question.
Yeah.
I always want to ask this question, but it's best for me to ask it here. How did animals, like land animals, like the turtles, get to the Galápagos Islands? And my second question is, during the expedition in 05 and 06, you say you collect the specimens. Are they shot and killed and bring it back dead? Okay, thank you.
Yes, I'll answer the second one first, and I'll say that yes, in 1905 and 1906, the birds were mostly shot, and then they were stuffed, and then they were put in the collections, and that was a very traditional way that ornithologists did that kind of work in the 1800s and early 1900s, before we had great optics. And, before we had other tools for studying birds, they might have had some mist nets, but they weren't that good. And even mist nets won't catch everything. So birds that fly up high, or seabirds that, don't come that close to you. They're very hard to catch in mist nets. So traditionally, folks used, guns for that. And for small birds, it would be a 410 shotgun, a small gauge shotgun. And based on some CT scanning we did of some of these birds in the collection, we think that they probably were loading the guns with sand. So we still do collect a lot of specimens in the collection, but most of what we add to the collections today are what we call salvage. So it's roadkill, window kill, things the cat dragged in, stuff we get from the local wildlife hospitals, stuff that washes up on the beach. And that's 90% of what we have in the collections. Every once in a while, we'll do some work in a very, in a totally unstudied area. When I first came here, we did some work in the Galápagos Mountains with colleagues at the Kunming Institute of Zoology to survey places that had never been surveyed before. And in those places, they did want to, collect full specimens because, again, it is the most complete and tangible record of what, of life on Earth. And when we collect these things today, we make sure that we, have full genetic material. We get RNA and DNA. We get stomach contents, and we collect as much as we can because we realize, we don't want to have those kinds of impacts. So I'd say that, yes, that is how they did things back then, but it's pretty rare that we do that now, today. We really have to justify it and have good reasons for doing it, but we do, some of that still today. The other question, and it's very important, I think, the work that we do and how many scientists use the specimens that we have. I did some work in New Guinea, and we collected off the islands from the southeastern tip of New Guinea to the Solomon Islands, and I would say that that's not necessarily the funnest work because it is hard if you love birds to have to collect some of them and put them in the collections. But after I got back, it was amazing how many people all over the world requested material from those specimens and used those specimens. And I've been a co-author on dozens and dozens of papers from that work. So, they are really important and they do a lot of good. The other question was, how did the tortoises get to the Galapagos? And lots of other specimens or species too. How did they first get to the Galapagos? So on the mainland, there are often big storms that wash trees down big rivers and out to sea. And on those plants, they sometimes call them rafts, and that means of dispersal is sometimes called rafting. But there's often lizards on those trees, and there's often plants on those clumps that get washed out to sea. And if you're on a sailboat out at sea, sometimes you'll pass these things floating by. So this does actually happen, and even though it's a rare event, over millions of years, things do hit the land. And the other interesting thing is that people now believe that there probably were giant tortoises on the mainland, but that they were probably eaten by humans and driven extinct by humans. So if you look at the closest relatives of the giant tortoises on the Galapagos and what's in South America now, there's evidence that there were closer relatives that were larger. The other thing that's interesting about the tortoises is that they do float. And there's evidence of sailors, sometimes they would collect more than they could use, or they'd collect a whole bunch, but then they ended up, and they would keep them on the ships for food. But then the whaling vessels, when they would catch whales, then they would throw some of the tortoises overboard. And some of those tortoises would wash back up on different shores and in different places. So we know that even the tortoises, even when they're not on a raft, they float. And they can go long distances at sea. And then it's just a matter of, do the currents deliver them to the place that they need to get? And if you think about the large tortoises on Alhambra Island and some of the even more remote islands out in the Pacific and Indian Ocean, it's even more mind-boggling that the tortoises could have gotten that far, but they might have. Darwin also did some really interesting experiments when he was on the Beagle. So one of the things is he wondered, he's like, I wonder how plants get out here. And so when seabirds landed, he did some experiments where he had some sterilized soil on the ship, and then he would rinse off the legs of seabirds and collect that water and put it into these pots to see what could grow and if anything could grow. And he did document that there definitely are plants that, birds carry on their feet. So that's another way that plants can get moved long distances as well by seabirds and other things. And of course, birds can fly. Some insects can fly, but tortoises not so much. They don't fly. Yeah, great questions.
If Jack has time, I think we have time for one or two more questions.
There's a question up here in the front.
Okay, thank you.
indicate that there was not, I don't know, the islands have not been affected by climate change as so many other places have?
Yeah, no, they definitely are being affected by climate change. I was when I was in the islands in October and in February, there's been a prolonged El Nino year there. And usually the water temperatures around the Galapagos are like between 60 and 70 degrees. And 60 degrees is pretty cold water. The bay is not that much colder than that. So it's really cold water. And when I was there in February, the temperatures on Santa Cruz were 80 degrees. And so the marine folks are really concerned about that. Now, because it's colder water, they don't have many coral reefs, right? There's very few corals because the water isn't that tropical, reef building coral, but there still are a lot of fish, and there are a lot of, other things that are expecting and relying on cold water. And when they don't have that cold water, the fish don't do as well. And then the seabirds don't do as well. So right now they're documenting a lot of seabird die-off during the El Nino. But the El Nino is also a cyclic thing, and we don't know as much about how long that used to happen. But some of the first big El Ninos were documented in the Galapagos in the 1970s, and some of the classic work done on Galapagos finches by the Grants. And there's a book about that called The Beak of the Finch by Jonathan Weiner, which is a Pulitzer Prize-winning book about the Grants' work. Because everyone thought that you couldn't see evolution, that evolution happened so slowly that and you've all seen pictures of the ancestral horse that had many toes, and then the horse today that only has one hoof, and how over 60 million years it progressed from here to here, and you'd think, oh well, in my lifetime I'm never going to see that. But the Grants worked on the small island of Daphne in the Galapagos, and it's small enough that they could band every bird on the island. And then and they weren't expecting this. They didn't know about El Ninos, but then when El Nino hit, they saw a huge die-off, and like 2/3 of the finches on the island died from the El Nino. And they actually saw a shift in the mean in exactly the way that Darwin says natural selection works. That the ones that can't make it get selected against. The two species of Darwin's finches shifted away from each other so that this one could specialize on large seeds and this one could specialize on small seeds. And it was like this perfect experiment. And then even after the El Nino was done, they stayed that way for some years because, those genes had been weeded out of the population. And so, even back then they were noticing these climate fluctuations, and they were noticing that it was, it had huge impacts on the species there. So the real question is like, as these changes, they've always gone like this probably over time, but now we're seeing them go like this. And, how that's going to affect things in the future, we're not 100% sure about. But climate change is definitely affecting the Galapagos. And how it's going to affect, we're not sure.
One more question?
Are interspecies hybrid offspring always sterile or sometimes not? And is that another vector for new species?
That's definitely a new vector for new species. And it's a great question. So like we have this idea for birds that, species don't interbreed, and in the biological species concept, which is defined as species that are actually or potentially interbreeding. And if they can't interbreed or they don't interbreed, then they're different species. Talk to any plant person and they'll tell you that's, that just doesn't apply to plants. Plants are so much more complicated than that. And you do often get hybrids. And, people who study orchids love to make new species or new hybrids of orchids. So life is much more complicated. And then when you get species that are in the process of speciating or that are close relatives, like between the two islands, or like the ground finches, we have the small ground finch, the medium ground finch, the large ground finch. But again, going back to Peter and Rosemary Grant working on the island of the island of Daphne, they documented during the time that they were there of other species of groundfinch coming onto the island and then breeding with the species that were there and then having offspring. And it added that variation to those species that then natural selection could play with. And then he was able to document, or not he, but they, Peter and Rosemary Grant, were able to document how this new influx of genes caused this complex thing to happen and added to the variation. So there's one ornithologist, and his name is Robert Zink, who periodically will publish a paper, because he'll argue that there aren't really 13 different Darwin's finches or Galapagos finches because they're all interbreeding. There's a lot of complex variation, but and there are other people that really would like to study that more. And when you look deep enough, you do see that they're, especially in the finches, which is one of the reasons why the finches wasn't the source of Darwin's breakthrough, because they're just too complicated. And there is this interspecies, and it's rare, but it does happen. And it is some of the stuff that contributes to real evolution. So, yeah. So we like to pretend that nature is really simple and fits in these boxes nicely, but it doesn't always.
Well, thank you so much, members, for joining us. Another round of applause for Jack.
Thanks very much.
And if you're interested in the Academy of Ventures, we do have some pamphlets up here. Have a great rest of your day, y'all.
And I'll stick around for a few minutes if you have any other questions or want to chat.